morning, children. Welcome back to our online classes. Today also we'll continue with the second chapter, that is sexual reproduction in flowering plants. Today we will start with post-fertilization structures and events. The major post-fertilization events include development of endosperm. It includes development of endosperm and embryo, maturation of ovules into seeds and ovary into fruit. They take place soon after the double fertilization. Now let us study about the endosperm development or development of an endosperm. Endosperm development precedes embryo development. That is, before the embryo development takes place, the endosperm development takes place. And the process takes place by different steps. The first step is primary endosperm cell. This one, primary endosperm cell, divides repeatedly and forms a triploid endosperm tissue. The cells of this tissues are filled with reserved food material and are used for the nutrition of the developing embryo. Second step is primary endosperm tissue undergoes successive free nuclear division to give rise to free nuclei. At this stage, it is called free nuclear endosperm. Third, wall formation takes place from the periphery and proceeds towards the center, and the endosperm becomes cellular. Now, seed formation, the endosperm nucleus, that is the triple nucleus, divides repeatedly and forms the endosperm in endospermic seeds. This endosperm acts as a food storage for the developing seed. Example is the maize. Okay, these are the three steps. This is the endosperm formation or modes of endosperm formation. Let us study the development of an embryo. The first stage in the development of plant zygote is a predetermined mode of development which is known as embryogeny. It gives rise to an organized mass of cells called the embryo that has the potentiality to form a complete plant. Most zygote divides only after certain amount of endosperm is formed to assure proper nutrition is provided to the developing embryo. In the majority of angiosperms, the zygote, that is the fertilized egg, divides by an asymmetric mitotic division and generates two cells with two different phases. The smaller daughter cell with dense cytoplasm is situated towards the chalazal pole side. It is called terminal cell or apical cell or embryonal cell. Another comparatively larger daughter cell situated towards the micropylar cell is called the basal cell. This cell divides transversely and gives rise to suspensor cells. The zygote gives rise to the proembryo and subsequently to the globular, heart-shaped and mature embryo. Though the seeds differ greatly, the early stages of embryo development, that is embryogeny, are similar in monopods and dipods. Here you can see zygote. It divides mitotically and produces two cells. Okay. Cells towards micropyle, embryo or terminal cells. and other are the basal cells or suspensor cells. Further, transverse division takes place and 6 to 10 suspensor cells are formed. Okay, here in the suspensor cells, there will be transverse division which will result in formation of 6 to 10 cells of suspension. Terminal cell of the suspensor is swollen. Okay, and it is vascular hostorium. Lowermost is the hypophysis to root tip and root cap. Right, so this stage is the globular stage. This is hypophysis. Okay, wherein you can see that here this is the heart shape, heart stage, wherein heart shapes structure is being formed, and then later it produces the mature embryo. This is mature embryo, which has the cotyledon, which has the root meristematic tissues. Now, embryo cell, that is vertical division at right angles to one another, and 4 cells, then transverse division, 8 cells, terminal 4 to plumule, and towards hypophysis to hypocotyle and radical. Proembryo is undifferentiated, globular, to heart-shaped, and then maturity. Now let us study the structure of a dicot embryo. A typical dicotyledonous embryo consists of an embryonal axis and 2 cotyledons. The portion of the embryonal axis above the level of cotyledon is the epicotyle, which terminates with the plumule or stem tip. The cylindrical portion below the level of cotyledon is the hypocotyle, that terminates in the radicle or root tip. The root tip is covered with a root cap. Example is mango, apple, radish, rose, etc. Here you can see this is the structure of a dicotyledonous embryo. Okay, wherein you will find 2 cotyledons. This is one cotyledon, this is second cotyledon. The portion above the embryonal axis, that is above the level of the cotyledon, is known as plumule. Okay, and from the plumule in future, the shoot will be developed, or it is called stem tip. The cylindrical portion, you can see here, the cylindrical portion below the level of cotyledon is hypocotyle, which terminates in radicle, or it is also called the root tip. And from the radicle in future, the roots will be developed. The root tip is covered with a root cap. Okay, so this is the structure of the dicot embryo. Let us study the structure of monocot embryo. Embryo of monocots possess only one cotyledon at one end, that is at the lateral end of the embryonal axis, and this cotyledon is called scutellum. In monocots, the cotyledon is known as scutellum. At the lower end, the embryonal axis has a radicle and root cap enclosed in an undifferentiated sheet, which is called coleorhiza. The portion of the embryonal axis above the level of attachment of scutellum is called the epicotyle. Epicotyle has a shoot apex and a few leaf primordia. enclosed in a hollow foliar structure called coleoptile. Example is grass, banana, bamboo, palm, etc. So you can see this structure. This is the structure of a monocotyledonous embryo, okay, wherein the scutellum is present on the lateral side. Okay, the scutellum is present on the lateral side. So this is what scutellum. At its lower end, the embryonal axis has the radicle. Okay, so this is the lower end, and at the lower end, the embryonal axis has the radicle. This is radicle, right? And radicle is covered by the root cap, and the radicle is then covered by the undifferentiated sheet, which is called polyorhiza. The portion above the embryonal axis, above the level of the attachment of the scutellum, is known as the epicotyle. And epicotyle has the shoot apex, right? This is epicotyle and it has the shoot apex. And a few leaf primordia are there. And it is covered by the undifferentiated sheet, which is called coleoptile. I hope this is clear to everyone. Now let us see. This is the endosperm development. Embryo, this is what? Endosperm, which stores the food for the developing embryo. And this is embryo where the plumule, radicle and cotyledons are present. If all the endosperm is absorbed by the developing embryo, the seed is a non-endospermic seed. Example, the broad bean, pea, groundnut. We'll be studying this in more detail. And if all the endosperm is not absorbed by the developing embryo, the seed is a non-endospermic seed. Example is maize, wheat, barley, castor. This is the structure of the dicot and monocotyledonous seed. Now, after fertilization, there will be different changes taking place in the ovary. Okay, in the ovule, the integuments which are there will get converted into seed coat. The micropyle will form the seed pore. Ovules will get converted into seed. Ovary will get converted into fruit. And wall of ovary is known as pericarp. Fruit development takes place from the ovary. The floral parts, the thalamus, also contribute in the fruit formation, such as in apple, strawberry, and cashew. And such fruits are developed. Some of the fruits are developed without fertilization. Example, banana. They are the seedless fruits. Double fertilization in angiosperms triggers the formation of ovule into a seed. The seeds are formed inside the fruits. Let us see the structure of the seed. A typical seed consists of a seed coat, cotyledon, and an embryonal axis. The seed coat is often double layered, formed by the integuments. The outer integument forms the outer seed coat or testa, which is hard, and inner tegument forms the inner seed coat or tegmen. The cotyledons' embryonic leaves are generally thick and swollen with food materials. These are 1 to 2 in number and rich in reserve food materials. Micropyle remains as a small opening found on the seed coat which facilitates the entry of water and oxygen into the seed. As the seeds mature, its water content is reduced and seeds become relatively dry. 10 to 15% moisture by mass. The hilum marks the point of attachment to the stalk. Sometimes the general metabolic activity of the embryo slows down and it may enter into the state of inactivity called dormancy or seed dormancy. When favorable conditions are available, this seed germinates into plant. Now there are different types of seeds which we have already completed, but let us do it once again. Depending upon the presence or absence of endosperm, the seeds having copious amount of endosperm tissues are called albuminous seeds, that is wheat, maize, castor, coconut, sunflower, etc. and seeds in which the endosperm is used up are called non-albuminous seeds, or they are called non-endospermic seeds like the beans, pea and groundnut. In some seeds, remains of nucellus are present called as the perisperm. Example in black pepper and wheat. The monocot and dicot seeds are named due to the number of cotyledons they have in the seed. Now there are different advantages of seeds. First, they are the basis of our agriculture. Second, the stored food in seeds supports the growth of seeds till they become nutritionally independent. Seeds are adaptive for seed dispersal to form new plant colonies. This can be stored for longer time as food source. Example, Lupinus articus of Arctic tundra, date palm of King Herod's palace near Dead Sea. Seeds are the products of sexual reproduction, so they provide genetic recombination. Now, in our next video, we'll be studying about formation of a fruit, significance of fruit formation, types of fruits, and some special mechanisms of reproduction. That's all for today. I hope you have understood the topic. Thank you.